Welcome to Brain Ponderings. This is going to be a special episode where I talk about our current understanding of what happens in the brain during aging and how we might counteract that through certain lifestyle and other interventions. By the way, if you like this podcast, please subscribe, and also if you have any comments on how to improve it or particular guests, then this podcast is oriented, as you may have gathered if you've listened before, to people with at least some basic background in cell biology, biochemistry a little bit would help too. But we try to make it so that overall, each episode, there's a lot of take-home messages that most anybody can get from the episodes. So brain aging and how to counteract it. This is something that most of my career, when I was at University of Kentucky, then as a lab chief at the National Institute on Aging for 20 years, is something I've focused the work on, the various students, postdocs, and other senior scientists in the lab. I started out as a developmental neurobiologist, and during my postdoc work, I was studying how neurons grow their axons and dendrites during brain development, how synapses form, and I discovered that neurotransmitter glutamate plays a major role in that. And then I got interested in nerve cells dying because it turns out neurons can be killed by high levels of excitation by glutamate. And we think this occurs during aging and in Alzheimer's, Parkinson's disease, for example. This is a quote here from the Nobel Prize laureate, Francois Jacob. It's truly amazing that a complex organism formed through an extraordinarily intricate process of morphogenesis should be unable to perform the much simpler task of merely maintaining what already exists. And by the way, so I'm showing slides on YouTube. So if you're listening on Spotify or other audio outlets, and you may want to, some point, if you're interested, look at the slides and actually what I'm showing. Okay, so everybody knows this, that during aging, our senses decline in their performance. Hearing, for example, a while back, my daughter was on the internet and she had found this hearing test, which tests your ability to hear different frequencies. And it turns out with high frequencies, she could hear them, I couldn't. And turns out that your high frequency perception of sound declines during aging, and that occurs in everybody, but in some it can be accelerated. And that has a lot to do actually with the sensory cells in your inner ear actually degenerating. Sense of smell declines, vision. I'm kind of I'm getting towards 70 years old. My vision's still pretty good. I got to wear glasses, but I'm getting last year when I got my eyes checked, I'm starting to get like some pre-cataract clouding of the cornea. And even touch and taste decline during aging. Okay, so that has to do with the nervous system and the brain, because obviously all these inputs go into the brain is where your neural circuits enable you to perceive them. Okay, what about other aspects of brain function? It turns out that beginning actually by 40 years old, there starts to be a decline in your cognitive abilities and your processing speed, your ability of your brain to process information rapidly, in reasoning, in memory, different domains of memory, verbal memory, other kinds of memory. About the only thing that doesn't decline beginning in your 40s is your vocabulary. So that tends to be, unless you get some neurodegenerative disorder, maintained. So, we've been hearing a lot in this election about age and Biden's and Trump's cognitive declines, which are fairly obvious. If you look back even 5 years ago, certainly 10 years ago, at each of them, there's been a decline. It's more so with Biden. And actually, because I've been at the Aging Institute and understand what happens in the brain during aging and have seen it, my own opinion is that no one, say, over the age of 65, when you really get a sharp decline in these cognitive functions, should be running for president, maybe even make a law, no one over 65, even though they have a lot of information in their brain, their ability to learn new things and adapt to different intellectual challenges compromised. Okay, so this slide just shows some data showing individual people and over time their decline in reasoning, processing speed, memory. Now there is scatter. There's a lot of scatter. So there, for example, are a few people that get to be 80 that are almost as good as some people in their 40s, but overall there's a decline. And in science, it's important to understand this general thing. So there's some variability. And I'm going to talk about later on what might account for some of this variability. For example, whether the person exercised regularly throughout their life, whether they ate a healthy diet and had a healthy body weight, or conversely, were overweight and had diabetes, which will accelerate cognitive decline. Okay, so we and others in the field spent a lot of time trying to understand what actually happens in nerve cells and other cells in the brain during aging that may explain some of these functional declines and may explain predisposition to age-related neurodegenerative disorders like Alzheimer's, Parkinson's, stroke. And one of the things my lab focused on is dysregulated energy metabolism in neurons, the problems in maintaining their cellular energy in the form of ATP. So what we're talking here about is compromised function of mitochondria, the energy factory of the cell. That does occur during normal aging. It occurs much more drastically in affected brain regions in Alzheimer's, Parkinson's disease. So this is kind of a wheel thing where, in this case, in this review article we wrote on hallmarks of brain aging, we focus on dysregulated neuronal energy metabolism, and I'll talk about that later, and mitochondrial dysfunction. But there's many other changes that occur during aging, and all of these other changes, for the most part, will tend to compromise neuronal bioenergetics, compromise mitochondrial function. So free radical damage, damage to proteins, DNA, membrane lipids by oxygen-free radicals. That increases during aging, accumulation of these oxidatively damaged proteins. The inability of neurons to constrain their excitability and therefore calcium levels within normal limits. Remember, when a neuron fires, there's sodium and calcium influx. And it turns out that calcium, if it stays too high too long in the neurons, can damage them. We did a lot of work on that in relation to Alzheimer's disease. There's impaired ability of cells to respond adaptively to stress during aging. There's impaired ability of them to remove damaged and dysfunctional molecules and organelles. For example, there's accumulation of dysfunctional mitochondria. This has to do with impairment in a process called autophagy, which I've had a number of experts, particularly David Rubenstein, talk about autophagy and brain aging. Impaired DNA repair. Will Bohr, who's Niels Bohr's grandson and a friend and colleague of mine at National Institute on Aging, he did a lot of work showing that DNA repair is impaired during aging, and that may contribute to neurodegenerative disorders and brain dysfunction. I mentioned aberrant neural network excitability, overexcitability of neurons by glutamate, impaired inhibitory neurotransmission mediated by the inhibitory transmitter GABA. There's inflammation that occurs in the brain during aging, having a lot to do with the glial cells, particularly microglia. It's a kind of an immune cell that is involved in tissue injury and inflammation. Stem cell exhaustion. There are stem cells in some regions of the brain that can divide and give rise to new neurons. This has been heavily studied in the hippocampus. I've had experts talk about that in podcasts, and there is some evidence that during aging, the ability of these stem cells to divide and produce new neurons is compromised. And then there's a couple changes that occur during aging that it's less clear, for example, what their contributions is to cognitive dysfunction or other functional declines in the brain or neurodegenerative disorders. But there is some evidence that cellular senescence plays a role, and telomere damage, and particularly telomerase deficiency. I had Ron DePino, who was down at the MD Anderson Cancer Center. It's actually recently had a nice study suggesting a role for a compromise of this telomerase protein in Alzheimer's disease. Okay, so I just want to kind of quickly mention that the core neural circuitry throughout the brain consists of excitatory glutamatergic neurons that are connected by synapses. And so these are the neurons that connect circuits within brain regions, that make connections between brain regions, that make connections from the brain to the spinal cord. So ultimately, these glutamatergic neurons are controlling all of our behaviors, learning and memory, mood, anxiety, that type of thing, all of our sensory perception, all of our motor output, our body movements are controlled by these glutamatergic neurons in various brain regions. And then there are inhibitory neurons that use the neurotransmitter GABA. These are present in all brain regions, and they act only locally within the brain region. And their function is to make sure that after a signal between these excitatory neurons occurs, that then the neuron is quieted so that it can then be ready for the next transmission of activity. So the GABAergic inhibitory neurons are important in constraining neural network excitability within normal limits. Other neurotransmitters like acetylcholine, serotonin, dopamine. Those neurons are very few in number. They're located in discrete brain regions. They extend their axons throughout most brain regions, and they can affect brain function only by modifying the ongoing activity of the glutamatergic neurons. And I've talked about this in a number of review articles before, and including in my most recent book, Sculptor and Destroyer, which talks about glutamate, the brain's most important neurotransmitter. So if you're interested in how the brain works and how it develops, how circuits are constructed during development, learning and memory, plasticity in adulthood, what goes wrong in neurodegenerative disorders, Sculptor and Destroyer is, I think, a good place to start. There's an artist in what was he? Netherlands, I think, who William Ottermolen, and he drew self-portraits over a number of years as he was developing dementia. Some 1967, well, and then actually the key thing here is 1996 when he first started to show really clear signs of cognitive impairment. And then over the next number of years, you can see the decline in his drawings to get to the point where you really can't tell this is a face at all. So as most of us know, Alzheimer's and Parkinson's are big problems because our population is getting older. We are all at risk for these disorders. There are some genetic factors that can affect our risk. There are environmental factors, which I'm going to talk about, that counteract aging and they protect against these disorders. So anyway, these have a big burden on individuals, loved ones, and society, a big monetary cost to society. That's shown down here. With Alzheimer's, it's estimated that it costs $600 billion in healthcare, and of course, people with dementia, the last few years of their life, they're going to need constant care. Okay. Now, most cases of age-related neurodegenerative disorders, there's no clear cause, so we call those sporadic. And then, late onset, they usually occur after the age of 60, 65, and most commonly, people with AD and PD become symptomatic in their 70s and 80s. There are very rare inherited cases of Alzheimer's, Parkinson's, also ALS, Lou Gehrig's disease, and Huntington's disease is a purely genetic disorder. But anyway, in the inherited cases of Alzheimer's, Parkinson's, and ALS, there's a gene mutation that causes the disease. If that person has the mutation, they inherited it from a parent, they will get the disease. They will get it at an early age, oftentimes in their 40s, 50s in the case of Alzheimer's, even younger in the case of Parkinson's and ALS. Okay, so most of us don't have these mutations. There's been a lot of work looking at the brains of people with these disorders, and what's become clear is in Alzheimer's, there's abnormal accumulations of amyloid protein outside the cell and tau protein inside the neurons. In Parkinson's disease, there's abnormal accumulation of a protein alpha-synuclein inside the affected neurons. In ALS, there's a protein called TDP-43 that accumulates. And we think this has to do with impairments in the autophagy system, the system that gets rid of these abnormal proteins and moves them to the lysosomes, this acid bath in the cell that degrades them. Okay, so here's the idea here. With aging in neurons, there's impaired bioenergetics, mitochondrial function, impaired calcium handling, as I mentioned, oxidative stress, impaired autophagy, inflammation. This promotes the accumulation of these proteins, amyloid, tau, TDP-43, alpha-synuclein. Also with aging, there's impaired adaptive stress responses, ability of cells to respond to stress. There's also reduced neurotrophic factor support. That is, growth factors that, like brain-derived neurotrophic factor or fibroblast growth factor 2, that promote the growth, survival, and synaptic plasticity. And so these things kind of get this feed-forward vicious cycle where you've got age-related changes that promote accumulation of these proteins. These accumulating proteins in turn cause stress on neurons, and so these kind of get this cascade and leads to synapse dysfunction, neural degeneration, and disease. I'm going to skip that. Just wanted to point out, we beginning in the early 1990s, together with Alan Butterfield, this is when I was at the University of Kentucky, we discovered that the amyloid protein, the amyloid beta peptide, it was known that it self-aggregates. And what we found is that when you add this self-aggregating protein to neurons, in culture, for example, hippocampal neurons, that's what we did a lot of work on, it accumulates on their membrane, and as it aggregates, it generates oxygen-free radicals that damage the membrane and impair the ability of the membrane to maintain the sodium and calcium gradients and restore these gradients after they fire. And one of my most important findings in this realm was that even low amounts of accumulation of amyloid beta peptide can make neurons very vulnerable to excitotoxicity, being overexcited and killed by glutamate. And we actually think that this excitotoxicity is killing neurons in Alzheimer's disease. It's well known that people with Alzheimer's have increased incidence of epileptic seizures. Evaluation of their neural network activity by imaging, EEG, and other methods show this hyperexcitability. We also did a lot of work on how the tau accumulates and think that there's a role for excessive calcium influx. Okay, I already mentioned this, but anyway, this is my book, Sculptor and Destroyer, and the different chapters, if you're interested in this. It's pretty interesting because, whether it's a cognitive disorder, Parkinson's, or a mental disorder like depression, like anxiety disorders, even schizophrenia, there's problems in these glutamatergic neural circuits. Okay, so how can you counteract brain aging? One way is regular exercise, particularly aerobic exercise. Studies in animal models, rats and mice, have shown that if you compare the brains of mice that run on running wheels every day to more sedentary mice that don't have running wheels in their cages, we find a number of things. For example, Henriette von Prague discovered that exercise stimulates neurogenesis, the production of new neurons from stem cells in the hippocampus, and that seems to play an important role in the ability of regular exercise to support and even enhance learning and memory. Interestingly, particularly a type of learning and memory called spatial navigation, remembering where you've been, what you saw, what you experienced. And this kind of makes sense from an evolutionary perspective that um, so we and other animals evolved moving through our environment and thinking and learning while we move through the environment. And so the actual movement and exercise as you move through the environment seems to enhance the ability of you to remember what you saw. And this could be a concern for people that I'm doing now and maybe you're doing now, sitting at a computer, not exercising. We still can learn a lot, but exercise bolsters cognitive function. I've found that. I think a lot of people who exercise regularly experience that their brain functions better, their mood is better, certainly. It has an anti-anxiety effect, which can actually help you focus too. Okay, so what else is going on in the brain? Well, the exercise seems to bolster the resistance of neurons to stress. So it can reduce oxidative damage, and there's even evidence that can suppress the development of the accumulation of abnormal proteins in experimental models that are relevant to Alzheimer's and Parkinson's. Interestingly, there's research that suggests that during exercise, proteins released from your muscle cells get into the blood and go to the brain and do good things to your neurons. For example, maybe upregulating brain-derived neurotrophic factor levels, promoting the formation of new synapses between neurons. Eiwu Cheng in my lab actually found that BDNF will increase the number of mitochondria in neurons, and that if she uses genetic tools to block mitochondrial biogenesis, BDNF is no longer able to promote the formation of new synapses or even to maintain existing synapses in the adult brain. So that's pretty interesting that physical exercise increases the number of mitochondria in neurons in the brain, and that those mitochondria then are critical for supporting synapses and neurons during aging. This is from a review article from Henriette von Prague and a postdoc in her lab. So if you're interested in this is a really good review article from 2018. If you're listening and not viewing, you'll have to go to my slides to get the reference here. So another thing that was found interestingly was that, and this is similar to what happens in muscle, that when you exercise a lot, there's actually an increase in the number of blood vessels in your muscle. That kind of makes sense because over time, as with regular exercise, your muscles get bigger, stronger, better endurance, and there's improved blood supply to provide the nutrients to support them and their function. But this has also been seen in the brain that physical exercise will increase angiogenesis, the growth of blood vessels, these small blood vessels, to supply more blood to the brain. Okay. And this is just another slide talking about exerkines, which is a term coined to describe proteins or other chemicals actually released from muscle, from liver, even from fat cells in response to exercise that seem to have beneficial effects on the brain. Here's a list of some of those: apelin, cathepsin B, fibroblast growth factor 21, GDNF 15 maybe, irisin maybe, interleukin 6, lactate. So people know about the idea that if you overwork, if you like exercise your muscle to exhaustion, there's lactic acid or lactate accumulation. It's called lactic acid threshold when your muscles have had too much exercise, intense exercise. But it turns out the lactate is a good energy source for neurons. And there's some evidence that lactate released into the blood during exercise can get to the brain and have good effects there. So pretty interesting research here. Okay, now I'm going to switch and talk about intermittent fasting, which is the first book I wrote, Science of Intermittent Fasting Revolution. We did a lot of work on this beginning in the 1990s, where we showed that if we maintain rats or mice on every other day fasting, and then look at the vulnerability of their neurons in their brains to dysfunction and degeneration in models that are relevant to Alzheimer's, Parkinson's, and stroke, we found that the intermittent fasting protected the neurons and lessened the brain dysfunction. And then we spent a lot of time trying to figure out what explains that, what's going on in brain cells that explains the ability of intermittent fasting to have such a robust neuroprotective effect. Well, so the way we define intermittent fasting is an eating pattern that includes periods of time of sufficient length or duration to cause a metabolic switch from your body and brain cells using liver-derived glucose, that is glucose in the liver. When that becomes depleted, which is about after maybe 6 hours in mice and after about 12 hours in humans, then what happens is when the liver glucose is depleted, fats are released from your fat cells. These actually particular fatty acids are medium chain triglycerides. And those then fatty acids are moved into the liver and converted to ketones. And those ketones then are used as an energy source. And there are two ketones, which I'll show you in the next slide. But anyway, the point is that when we do experiments in human studies with intermittent fasting, all of the intermittent fasting eating patterns have been shown to cause this metabolic switch and elevation of ketones during the fasting period. And I'll talk about it. The two most common ways of doing intermittent fasting in humans are daily time restricted eating, constraining the time window you eat each day to say 6 to 8 hours. So you're fasting, 16 to 18 hours every day. Another one is called 5-2 intermittent fasting, where two days a week you eat only about 600 calories. And that's not enough to prevent this metabolic switch and the elevation of ketones. Okay, so in this slide that shows a cell, and what we found, and others have worked on this a lot too, is that hydroxybutyrate, beta hydroxybutyrate, it's moved into neurons, and it can be used as a source of producing ATP in mitochondria. But it also has signaling functions. It activates a transcription factor called CREB, which is known to upregulate BDNF, enhance learning and memory. In fact, we did show that this ketone, beta hydroxybutyrate, stimulates BDNF production in neurons and mitochondrial biogenesis. So it's kind of interesting, this ketone, which now, if you think about this from an evolutionary perspective, you're an animal or a agricultural revolution hunter-gatherer ancestors, and you haven't found food for a day or two or three, the ketones are up, and it kind of makes sense that these ketones not only provide an energy source, but they've evolved to enhance the functionality of the brain as you're trying to find food. You don't want your brain to not work well when you're starving. Okay, so this looks a little more detail about this, what's happening with this metabolic switch. Okay, with a certain amount of fasting, 12 hours in humans, for example, and that's if you don't exercise. If you exercise during the fasting period, that will accelerate the onset of the metabolic switch, and you'll get elevation of ketones quicker. But anyway, so fasting, you get glycogen depletion, that's essentially glucose, and then the fat cells release these free fatty acids into the blood. They're moved into the liver cells, converted to the ketones, BHB and acetoacetate. These are actively moved into the blood, out of the blood, into cells, including neurons in the brain, where they do good things. In fact, there's clinical trials now of giving this beta-hydroxybutyrate in patients with mild cognitive impairment or early Alzheimer's disease. And so I'm excited to see the results of those. Actually, one of the studies is by Dimitrios Kapagiannis is heading it. I had him on a recent podcast, and he did a nice study on showing that intermittent fasting is beneficial for the brain in older individuals at risk for cognitive impairment. Okay, so that's that. Okay, so back in the 1980s, my friend and colleague Don Ingram and his co-workers at the National Institute on Aging showed that if you maintain rats or mice on every other day intermittent fasting, beginning when they're young adults, and you maintain that throughout their lifespan, they live twice as long as the animals that are fed ad libitum, that is, that have food available every day, all the time. And these animals, on intermittent fasting, they have muscle mass similar to the control animals, not on intermittent fasting, but they have lower body weight because the animals who aren't on intermittent fasting are accumulating a lot of abdominal fat and some subcutaneous. And that makes sense because the intermittent fasting, on the fasting days, the animals are using up the fat that's been stored in the eating day. Okay, I mentioned in various models, we found that intermittent fasting is beneficial for the brain. This is just showing some examples from using an epilepsy model where we give the animals an excitotoxin, a chemical that excessively activates glutamate. It causes epileptic seizures and death of neurons in the hippocampus and cognitive impairment. And we found that the intermittent fasting suppresses seizures and lessens the brain damage, lessens the cognitive deficits. And in fact, the ketone ester, the ketone that's being tried in the clinical trials, is also beneficial in suppressing seizures. In a toxin and a monkey, actually a monkey neurotoxin model of Parkinson's disease, we found that daily calorie restriction is beneficial. In a rat stroke model, intermittent fasting was neuroprotective, as well as in a Huntington's disease model. These are results from an Alzheimer's model. This is a mouse model that Frank LaFerla developed with our help. more than 20 years ago. These mice have mutations in 3 human genes, that 2 of which cause early onset Alzheimer's disease. That's a mutation in tau and a mutation in the amyloid precursor protein. And the other mutation is a mutation that causes frontotemporal lobe dementia. Sorry, that's the tau mutation. And then we had a presenilin mutation that caused that early. Anyway, the bottom line is, as these mice age, they accumulate a lot of amyloid plaques, shown here in green, A beta plaques, and intracellular tau and cognitive deficits. And this slide just shows that daily time-restricted eating with calorie restriction, or every other day fasting, when these animals are young, will largely prevent, at least at the time point we looked at in 17-month-old animals, essentially prevented the cognitive impairment compared to control non-Alzheimer's mice. Oh, yeah, here's some slides from Dr. Kapagiannis' recent study in cell metabolism, where he had 40 people between the ages of 55 and 70 with obesity and insulin resistance, which is a precursor to diabetes. So we know these people are at risk for cognitive decline. There's a lot of evidence for that, and for Alzheimer's disease. So they were randomly assigned to either 5-2 intermittent fasting, so 2 days a week, eating only about 600 calories, or the USDA healthy living diet, which is generally it's rich in bottom of the food chain, so it's largely plant-based, and tends to have, people who eat this tend to have a lower calorie intake because you get a lot of bulk in your stomach, I guess. But anyway, so before they were started on these diets, a lot of data was collected. Cognitive testing, measures of glucose regulation, a lot of blood markers. What else? Magnetic resonance imaging, looking at both brain structure and neural network activity in their brains, and then isolating extracellular vesicles, which we talked about with Dimitrios, which are these small vesicles released by neurons and other cells. They get into the blood, and Dimitrios developed a method that you can isolate vesicles that are coming from brain neurons from blood and interrogate them. So he looked at tau, and he looked at a marker of neuronal insulin resistance, and they looked at amyloid beta peptide and so on. Okay, so I'm just going to summarize the data. The participants in both diets lost a significant amount of weight over the 2 months. Typically about 2% of their initial body weight. Their fasting blood insulin and glucose levels were significantly reduced, a little bit more so with the intermittent fasting compared to the healthy living diet. The ketones, as you would expect, were up more in the intermittent fasting group. And so, yeah, so that's both beta hydroxybutyrate and acetoacetate. Total cholesterol was down a little bit more so with the intermittent fasting, as well as LDL levels, the if you will, bad cholesterol. Then they looked at a variety of measures of cognitive function and found that several were improved with intermittent fasting, and somewhat with a healthy living diet, including executive function, fluency factor, and this dimensional set shifting, which is your ability to switch your attention and focus to a new task, and then a number of different verbal learning and recall tests. So you try to memorize, you're given a certain number of words, and then there's a time interval, and then they test you to see how many you remember. So yeah, so there's an improvement in learning and memory with intermittent fasting in particular. And then the measures of brain insulin resistance were somewhat reduced. And there's this brain age method of a kind of semi-quantitatively assessing the relative age of the brain. And that seemed to be reduced over time in pretty much all of the subjects, individual subjects. The cerebrospinal fluid measurements of amyloid and tau, there's really kind of no clear significant differences. Now, I had a graduate student, Alexis Stranahan, who, it's been well known, more than 15 years ago. She took normal mice and diabetic mice. They're genetically diabetic because they have a mutation in the leptin receptor and don't respond to this stop eating hormone that's released into your blood when you eat, when you start to get full. And by the way, a lot of people with obesity have leptin resistance, so they perceive that they're hungry when other people without obesity would not. But anyway, so she took normal mice, diabetic mice. She randomly assigned them to the following conditions: ad libitum feeding, sedentary, that is, no running wheels in their cages; ad libitum feeding, running wheels in the cages; daily calorie restriction with time-restricted eating, sedentary, and then the time-restricted eating, calorie restriction, running. And then after 3 months, she took out their brains, and in one hippocampus, say the right hippocampus, she quantified number of synapses on neurons in a particular region of the hippocampus that's critical for spatial learning and memory. And so she counted synapses. And then the other hippocampus, she measured levels of BDNF. And what she found is that both exercise and intermittent fasting, in this case with calorie restriction, increased number of synapses, increased BDNF levels. She also found that the diabetic mice had fewer synapses to start with and lower BDNF levels. However, both the exercise and the intermittent fasting increased synapse numbers and BDNF levels in the diabetic mice. And there was a additive effect where the combination of exercise and intermittent fasting resulted in the greatest increase in number of synapses and BDNF levels. Now, before I finish up, I want to talk about one particular protein that before I retired, we were really doing a lot of work on. I think some of the best work we've done. And that has to do with a protein called SIRT3, SIRT3, SIRT2 and 3. And it's in the mitochondria only. That's the only place it is in cells, including neurons. And it controls the function of hundreds of other proteins in the mitochondria. And the way this SIRT3 controls their function is by a process called deacetylation. It removes a certain molecule called an acetyl group acyl group from proteins. And when it removes that molecule from the protein, it can affect their functions. For example, Wu Chang finds that sirtuin-3, by deacetylating an antioxidant enzyme called superoxide dismutase 2, increases the antioxidant defenses in the mitochondria. She also found that the SIRT3 affects a protein in the membrane of the mitochondria that stabilizes the mitochondria and makes it more resistant to stress. And then by studying mice lacking SIRT3, Awu, as well as Yang Lu in the lab, and others working together with them on this, found that SIRT3 plays a critical role in the ability of neural networks to respond to intermittent fasting. So in these SIRT3 knockout mice, intermittent fasting is no longer able to suppress neural network excitability and protect the neurons against excitotoxic stress. So that's pretty interesting. We also had some evidence that this SIRT3 in mitochondria, interestingly, has an anti-anxiety function. And we didn't really figure out in detail how all that's working, but it could involve ketones and somewhat, because ketones, of course, are moved into the mitochondria and may affect SIRT3. So this is pretty much my last slide here before I mentioned one other way besides exercise and calorie restriction, intermittent fasting to protect the brain against aging. So the idea here is this switching between fasting or exercise or the combination and recovery, eating, resting, sleeping. The switching back and forth over time is good for the body and brain. And we have an idea what's going on in neurons during the challenge period, the fasting challenge, the exercise challenge. We have an idea of some of the changes that are occurring during the recovery period, eating, sleeping, resting. And one general thing that's occurring is that during the challenge, the bioenergetic challenge, the fasting or exercise period, cells go into a conserve resources, stress resistance mode. So that has to do with autophagy and bolstering whatever antioxidant defenses. Then during the recovery period, cells go into a growth and plasticity mode where, for example, the mTOR pathway, which is upregulates protein synthesis, is turned on. So that makes a lot of sense. So after you've chased your prey for an hour and killed it, and of course this is after you haven't eaten for several days or a week, and then when you eat your food, your cells are geared up to take up amino acids, to take up other nutrients and use them. And those pathways are turned up in response to the fasting and exercise, so that when you do eat and rest and sleep, the cells are able to grow, get stronger, and more resilient. The last thing I want to mention is the third way to counteract brain aging, and that is to do what you and I are doing now, is to engage in intellectual challenges. Keep your mind intellectually challenged. Learn new things. Push yourself to learn new things. Perhaps you'll listen or watch this podcast and say, hey, maybe I'll read a review article that Mark Mattson's been talking about, and I'll put some in the description section. And by the way, use PubMed. That's the resource for scientific biomedical research. And if you go on PubMed and you enter the search terms exercise, brain, review, use the word review, get review articles. Maybe you put neurogenesis if you're interested in that. Then you'll get a review article, and that's the place to start. So use PubMed. Now, it turns out if you raise mice in what's called an enriched environment, where they have big cages, they have a lot of animals in the cage, they have places to hide, to build nests, things to climb on, and so on. Their brains are healthier, their cognition is improved, and during brain aging, there's a deceleration, I guess, if you will, in the rate of brain aging. So there's been a lot of work on environmental enrichment in the brain. It turns out there's roles for brain-derived neurotrophic factor, neurogenesis, synaptic plasticity, and some of the same effects actually of exercise and intermittent fasting that are occurring in these neural networks. Yeah, and so this is from a review article by Gerd Kemperman a few years ago in Nature Reviews in Neuroscience on environmental enrichment, that is intellectual challenging lifestyles and the brain in regards to health and disease. And there's a lot of information in this article I'm not going to go into now. In terms of mechanisms, a lot of overlap, as I mentioned, with exercise and intermittent fasting, and a lot of implications for the clinic. I should also mention, I've talked a lot about mechanisms, which a lot of those are done in animal studies, but there's many, many studies showing that regular exercise is beneficial for the brains of humans, not only in improving mood, but cognition and protecting against cognitive decline, Alzheimer's disease, probably Parkinson's disease as well. Same with intermittent fasting. Fewer studies looking at the brain with aging with intermittent fasting, but a lot of evidence that intermittent fasting causes systemic changes that benefit the brain, improve glucose regulation, reduce blood pressure, and others. Okay, so I hope you've enjoyed this podcast. It's more kind of like a lecture presentation, and I thought this might be good. If you like this, make a comment on it. I could do this intermittently and then intersperse these kinds of presentations of information with my regular podcast with my guests who are scientists at the forefront of their particular area of brain research. Yeah, so do let me know. I'm teaching a course on brain bioenergetics and neuroplasticity soon at Johns Hopkins, and I could probably also record those lectures for you if you're interested in that. Okay, have a good rest of the day. Take care, exercise, don't overeat, maybe do intermittent fasting, keep intellectually challenged. Till next time, goodbye.